What's the difference between lytic and lysogenic? Okay. And remember, lytic is, for lack of a better term, the one-night stand. The virus goes in, breaks up, and then leaves. The other one is the long, steady relationship. Because how does it do it? It gets into the DNA of the chromosome or attaches itself to the membrane. Right? What's the difference between virulent and temperate? Virulent does what? Virulent only does this. Temperate does both. So temperate, both. Virulent, only this. Right? Lysogenic conversion. We talked about Vibrio cholera and Corynebacterium diphtheriae. Right? Both of them, remember, produce exotoxins. That's one way to link this to the last chapter. Both of them produce exotoxins. But the exotoxins are encoded by what? By the virus. So it's not actually on the chromosome itself. This is a case where the virus, the bacterium has to be sick to cause sickness. Literally. It has to be infected by a virus to be able to infect humans. Oh, lytic and generalized and specialized. Right? Know that generalized, again, transfers any, but at a low frequency. Specialized transfers only certain ones, but at a higher frequency. What are examples? I discussed lambda, and then please know the insertion sites of lambda. Lambda inserts between the biotin and galactose operons. So these are the only ones that can be transduced. That's specialized. Right? Those that break up the DNA and fill up their head, these would be generalized, because really anything can be transferred at a low frequency. The last part of this handout deals with prions and viroids. Again, the size is not important, okay? But these are the big ideas with viroids, real quick. In fact, there's a table about PUPC, viroidea, and don't worry about that table. Okay? Viroids, real quick. Viroids are RNA that cause plant diseases. They're very small, right? They need to replicate. They need to use the host, and they're transferred by plasmodesmata. So the big ideas: plant RNA. Prions. You get a little bit more questions on prions. Right? And remember, prions are proteins that cause animal diseases. Please know the diseases: Kuru, humans, right? Chronic wasting disease, deer, elk. Mad cow disease, cow. Creutzfeldt-Jakob disease, human. What's mad cow disease in humans called? Well, Creutzfeldt-Jakob, but it has N or NV, not new or new variant or variant, simply variant disease. That's another point right there. What's the difference between PRP-C versus PRP-SC? Right? So for example, so you might have a quick key on these right there. So for example, here's my key real quick. Okay, so here's the question. PRP-SC, PRP-C, both, neither. Which one of these is the abnormal protein? A, the scrapie is the abnormal. The normal is the cellular. Which one of these causes disease? PRP-SC. Which one of these is made up of amino acids? Oh, because these are protein, right? Let's see. So one would be, which one would be neither? Which one of these causes disease in plants? Neither, right? Which one of these causes disease in animals? PRPS. Which one is predominantly alpha? Alpha helices. Okay. Right? The normal one. The abnormal one is beta sheet. Which one of these is protease sensitive? Makes sense, right? If it's protease sensitive, it's the normal one. That's why the abnormal one is that bad, because it really can't be, almost destroyed. So, any questions about this chapter or this handout? Now, the last chapter should be the freshest, and I honestly think it's the Well, of the two, with viruses, it's certainly easier, it's shorter, and it's not really complicated, it's a bunch of terms, okay? And you will be asked about these. So let me run down by the, quickly. First of all, signs and symptoms, for example. Fever is a sign of a disease, true or false? Based on our definition. Sign is observable, measurable, certain, right? Fever you can easily measure. Nausea you can't. So rash you can, etc. What is the difference between toxigenicity and, first of all, what's the difference between virulence and pathogenicity? Right? Pathogenicity is a qualitative, virulence is quantitative. Right? So you can have different levels. Did you guys sign the paper by any chance? Yeah, just, oh, you got it? Okay. If you haven't signed it, please sign. This is just an attendance sheet for their purposes. Thank you. Also, signs, symptoms. Oh, what's the difference between a frank and an opportunistic pathogen? And be able to give examples. Okay? So remember, an opportunistic pathogen is one that will only cause disease if given the opportunity. It's called secondary. Primary is called frank. True. It doesn't need help. It will cause disease no matter what. The example I gave, I think, is Shigella. There's a bunch of them. The steps of the disease, as I said, you will be asked that question. Incubation, prodrome, clinical, etc., etc. You will be asked to differentiate between, in fact, let me list some of the important terms.